Next up is Katie James. So Katie, if you want to get your slides ready.
Sure.
So Katie is an undergraduate at the University of New Mexico. And she will be talking about the contribution of anxiety to spatial memory deficits in the TgF344-AD rat model of Alzheimer's disease. So please go ahead.
Thank you so much. So as you mentioned, my name is Katie. I'm a sophomore at the University of New Mexico, and currently we're working through this longitudinal study in this rat model of Alzheimer's disease, looking at whether or not the magnitude of anxiety is a behavior, is a later predictor of spatial performance in these rats with AD. And just a note, this is kind of a snapshot of what we're working with. We're by no means, finished. So just some background on the topic. We see in Alzheimer's disease that anxiety-like behaviors and spatial navigation impairments are early markers. For example, in this figure here, we see that the husband in this partnership is easily able to navigate himself between home and the coffee shop, and his wife doesn't seem to be able to do that as easily. And this is something that we would see in someone with Alzheimer's disease, not quite knowing where they are in their environment. And in this rat model, TgF344, we see these anxiety-like behaviors around 4 months, and then the spatial deficits later at 7 to 10 months. So still early in their lifetime, but anxiety-like behaviors a little bit earlier. And just from time and time again in humans, we see the relationship between increased anxiety, having a negative relationship on working memory capacity, and more relevant to us, spatial working memory performance. And so all of this together led us to wonder, since these rats show increased anxiety-like behaviors early on, is this a predictor of their performance in the spatial working memory task? And we think that it will be. So to analyze their spatial working memory performance, we use the match to place task. So in this task, we have a Morris-Watermaze variant where the platform is in a specific location on day 1. They have 2 trials to find that platform. We expect that they would find it quicker on trial 2. And then we move the location every day for 5 days. And we do this across 3 time points from 4 to 5 months of age, 7 to 8 months of age, and then 10 to 11 months of age. To assess their anxiety, we use the elevated plus maze again at each time point, where time spent in the closed arm here would indicate higher anxiety, or at least what we think to be higher anxiety. Of course, the rat wouldn't be able to tell us if they're anxious or not. But, all in all, we used this simple linear regression where anxiety-like behaviors would predict spatial deficits. So looking at our results so far, we see that the rats adhere to what we would expect. Looking here at, age versus closed arm in seconds, meaning our anxiety measure, where more time spent in the closed arm is higher anxiety. We see that the TG rats are consistently spending more time in the closed arm across all time points. And then looking at their spatial performance across all time points, a different score normalized across both trials, where a negative difference score indicates finding the platform faster on trial 2. We see that nearly all of the positive difference scores are attributed to rats with Alzheimer's disease, and generally they're experiencing more spatial deficits where their scores are more positive, indicating worse performance. So this is what we would expect. We see that the TG rats show greater anxiety-like behaviors and show greater spatial deficits. However, when we plotted these together, where our anxiety measure was to predict the spatial measure, we don't see much of a relationship here. And so we conclude, based just on this, that the time spent in the closed arm was not a predictor of latency. However, we think that this might be because escape latency, as Matthew went over in his presentation, would be more of an indirect measure of the variance in the behavior. And so we want to, in our next step of the experiment or analysis, look at the direct analysis of their behavior in the form of swim trajectories. So This is a figure I made to show in part A here, the whole path of the rat broken up into different trajectories. Here in part B that we see it is executing, and then all the different kinds that we might expect. And for rats specifically with high anxiety, we might see them using paths such as thigmotaxis or chaining, where they're definitely indirectly navigating around the platform, or just kind of sticking to the walls of the platform, not really moving about, not confident in their ability to find the platform. So that's kind of the next step where we're heading, and hoping to see more of a relationship between the search strategy and the spatial performance in the rats with AD and anxiety. And so just a quick thank you to all the members in Ben Clark's lab, specifically Laura and Ryan, my direct mentors, and then the members of the Pankowski lab who helped us with the initial experiment. And I've also linked my science Twitter here. I am very new to science Twitter, looking to make connections, and obviously my email. And then a thank you to our funders. So that's all I have if there's any questions.
Great, thank you very much. Were you a bit disappointed that you didn't find the relationship you were hoping for?
Yes, obviously that's always kind of disappointing, but it's not a dead end for us, and that's something to look forward to.
Right. So can you tell me a little bit more about the TG model for Alzheimer's? So the rats, because I don't really know anything about them at all.
Sure. So so relatively new model, not as many papers on this AD model as others, but basically this is the one that we've used in our lab to analyze their performance in basic reference memory tasks. I don't know personally a ton about the difference between this model and others, as I'm fairly new to this, but this is just what we specifically work with.
Okay. No, I mean, Alzheimer's is a huge issue, and for society, if we can find ways of getting to better understand it, that would be awesome, of course. Absolutely. So I mean, I've heard ideas beforehand of this, using things like the raised maze that you're also using. But yeah, I guess you'd have to see whether using something like Matthew's software would be useful in something like the water maze instead. See if you can get better results from that.
Definitely.
There's a question, I think, in the Q&A from Jason asking, are you going to look at the brains? Maybe there are dorsal ventral differences in pathology that could explain the spatial anxiety dissociation.
I mean, definitely that's a possibility. We're kind of still in the beginning stages of this, and just kind of wanted to come here to report what we've found so far. I think it would definitely help us to look at more data, obviously, as we didn't find exactly the relationship that we were hoping for. So it's definitely a possibility, sure.
Okay. Do you mind going back to the results slide again? I just want to see it once more. Sure. That's okay.
That too, I'm not exactly sure.
Right, okay. Right. Yeah, I guess it's hard to really conclude much from that, right?
Right. The only thing that we really see when we look at this is that the TG rats really only exist in this upper range, as we saw from them having high anxiety. And we do see a good amount of rats here at the 300 seconds that just didn't move at all. Right. So that's something obviously to consider, that they may not have high anxiety, they just didn't move, which is always when working with the rats, when they don't move.
Easy rats, yeah. So they basically just spent the entire time in the closed arm, yeah?
Yes, the total trial was 300 seconds, so that would indicate they didn't move.
Okay. Yeah. Okay, well, I mean, yeah. I think it might be hard to conclude very much, I guess, based on this. But maybe you'll get more of a result with the water maze instead. Yeah. Any questions from the audience? Again, if you want to raise your hand, then we can also let you ask the question live, or you can write something in the Q&A. You're welcome to do that too. So do you mind going back to the final slide, your conclusion slide? Yeah, so this one here too. Okay. Yes, it's it's impressive the amount of different types of behavior that behaviorists can notice in a rat, where I just see a rat that's swimming in the water.
Sure.
But yeah, I'm guessing this is where something like Matthew's software could actually be quite useful for classifying this kind of behavior.
Yeah, I had a question about that. So you have a lot of strategies here, which is something we kind of like. Obviously, I don't know if you've started doing any preliminary analyses here, but we struggled a lot with trying to dissociate what like a true strategy that they might be employing was. And like, is there a difference between something like a target search and a target scan that you have here? Like, is that a different behavior that the animal is employing versus are you just dividing strategies? And it really does make a difference when you start trying to do statistical analyses on these data. We've worked with statisticians now trying to figure out good ways of analyzing the strategy data statistically. And really some of the best advice that they had was break it into spatial and non-spatial only and just do a bivariate analysis on that. So and it's kind of just a word of caution, and kind of maybe you have some insight of being very careful and trying to dissociate like true different strategies. Like thigmotaxis, the animals do behave this way, and it's different than swimming randomly in a maze. But and chaining might be, it's like a little more central thigmotaxis. It's probably, I mean, they might know the distance of the platform from the wall, but then you start getting into like, okay, now we have like focus search versus like a focal search near the platform. Are those different behaviors? Is one slightly? So I'm just wondering if you had any input on that, and maybe how far you've gotten into this.
So we definitely I've worked with, we have our own kind of in-house way to do this, but definitely have looked through your GitHub as well as we move into doing this more centrally. But that's kind of why I asked earlier about reliability, because, when we look at, as you mentioned, target search versus target scan, how different really are they? And so I know that maybe if I was manually, coding it, I would worry that I wouldn't treat the same path the same if I saw it twice. So I definitely, appreciate your advice on that. This was just kind of based on work we had seen before with the specific rat model. So that's kind of where it came from.
Matthew also had a question about the open arms. I don't know if you want to ask that still, Matthew.
Sure, yeah, I was wondering if you tried releasing the rats for your open arm, like in the open arm of the maze, because that would kind of allow you to see if they were super anxious. I'd assume they would just move to the closed arm and then stay there, rather than if you release them in a closed arm, then they don't move, and then it's harder to dissociate whether they're just being extremely anxious and not wanting to explore at all, or whether they're just lazy and are going to lay there for 5 minutes.
Sure. This experiment was done before I even came to the university, so that's, I didn't get the chance to ask at the time, but something that kind of has come up in that conversation is if the rat's not going to move, they're also not going to move in the open arm. So that would, like, the open arm and closed arm are just inverse numbers of each other, basically. So we might see the exact same thing.
Okay, we are getting close to the ending time for the whole session, unless there's a really burning question from anybody. No. All right. Good. Thank you, Katie.
Thank you.
And thank you to all the speakers for today. Very interesting stuff being discussed, of course. I'm afraid we do have to stop at this point right now. Just want to make sure to say thank you also to Siddharth Suresh, who's been sitting in the background, making sure everything is working okay. And yeah, I think this is pretty much the last session right now, except for the closing ceremony, so to speak, right after this. So I hope you have a continued good weekend, and I'm sure I'll see you around.
Thank you very
everyone.
much.
Thank you.
Thank you.